So the next couple of videos we'll be looking at cell division. This one will be focusing on mitosis. Next one we'll be looking at meiosis. Just a quick look at the outcomes covered. Okay, so cell division. So in unicellular protists, the common form of cell division is asexual binary fission. So in binary fission, we have one organism becomes two. Now there's no sex cells or gametes involved, it's just one cell becoming two. We could add to that similarly with single-celled yeasts with budding. We have a cell division where one organism becomes two. But if we're looking at multicellular organisms, cell division by mitosis results in two new identical cells that contribute to the growth of the whole organism. We're not making a new organism with mitosis, we're simply contributing to growth of the whole organism. Now, sex cells and gametes are not involved in mitosis, but we'll see next lesson they are involved in meiosis. So we'll talk about this distinction more, but basically mitosis is involved in all of our cells except for the gametes. So we get that's where we get our idea where mitosis contributes to growth of the whole organism, but it's got nothing to do with creating a new organism. That's only in the gametes. Gametes is only when we have meiosis. So just one little important thing we need to do when we are making new cells, we need to make sure that we copy the DNA correctly. So now obviously when a cell divides into two, we need to make a copy of the DNA. It's important that that copy is done to prevent mistakes that could be made, which would be mutations, which often would have a negative effect. So that's just keep in mind, that's a very important part of any kind of cell division. Now, in unicellular organisms, the DNA must be copied exactly from parent to offspring to maintain the genetic stability of the species. It's a bit more nuanced in multicellular organisms. So the cells that make up the body, we need the DNA to be copied properly so that those cells can continue to function in their proper way, so the proper and coordinated way that will help the whole organism. But importantly, any mutations that may occur in any of our body cells except for the gametes. So if we're talking about everything else that's involving mitosis, any mistakes or mutations involved will not be passed on to offspring. Any mutations or mistakes that are made when dividing cells, that will only happen if it's happening in the gametes. Yeah, and that just kind of shows you we're kind of rolling the dice a little bit on that, because I know that we learn with evolution that variation is a good thing. And yes, variation is a good thing, but usually if we have mutations in our cells, most of the time that means that we're actually making things worse. Our cells are kind of like very evolved. It's kind of like it isn't broke, so don't fix it. Most of the time we would think mutations in this context is actually bad. Now, so just looking at chromosomes, so we know that the DNA needs to be copied and divided between the two new cells. So in preparation for being divided between the two new cells, the DNA and the proteins in the chromosomes condenses to a very tight and compact form so that it's visible under a microscope. Now, keep in mind, the chromosomes are not usually visible under a microscope like this. It only happens during mitosis and in preparation for mitosis. And you could think of it as like the tightly packed chromosomes. It's like you're going to move house and you've packed up all of your stuff in boxes, kind of ready to move. It's only like that when you're moving. Once you've got to the new house, you're going to unpack everything up again. So just keep that in mind. We don't always expect to look at cells under a microscope and see chromosomes looking like this. It's only during mitosis. So we'll just need to know a few I guess to be able to label a few parts and talk about some parts correctly. So what we have here first is what we will see normally before before the DNA has been replicated or copied. So we'll have homologous chromosomes, and the idea with the homologous chromosomes is that for every chromosome in our body, we have one copy from our mum, one copy from our dad. So if this, for example, was to be chromosome 1, then we could just say, I don't know, the blue one could be from dad, the red one could be from mum. So it's the same chromosome. The two copies are slightly different, but we could say they're very closely related because they're chromosome 1, and we call that homologous chromosomes. Now there's a part in the center that's the centromere. That's kind of where, I guess the part where the chromosome is kind of held and moved later on, you'll see. Now, so when the chromosome is copied in preparation for cell division, we then get this kind of X-looking chromosome, and the X is kind of held together by the centromere. So one thing that's a little bit confusing though is when we're talking about chromosomes that have been copied and are in this X configuration, we're still calling them like homologous chromosome 1, just like we would call this single one homologous chromosome 1. This is still homologous chromosome 1. Now, the sister chromatids just kind of refer to the two different copies now that have been made. So what we see on the left would be one sister chromatid, on the right, the other sister chromatid, both of them on the same homologous chromosome. And this just showing that again, we've got individual chromatids because they're on the same chromosome, they're sister chromatids. We have the centromere, but just keep in mind, single chromosome, normally it would be like this. We only see this duplicated chromosome when mitosis is happening. On all of the other times, we would just be looking to see that. So what does this, I guess, look like in kind of real life? So this is some images that have been taken from a microscope. Now, the chromosomes aren't automatically nice and straight. This has kind of just been fixed up a little bit to make it easy. So this is what we would see with humans. So with humans, we have, there are 23 chromosomes, and for each of the 23, there is a pair. And so again, for that pair, we get one from our mother, one from our father. And they would all look identical except for the last one, which would the sex chromosome that determines gender. So now if we were looking at a female, we would see two identical X chromosomes, where one comes from the mother, one comes from the father. Or if we were looking at a male, we would normally expect to see one X chromosome and one Y chromosome. And again, one of these would be from the mother, and one of these would be from the father. And we can guess which one, because the mother would be XX. The mother can only donate an X chromosome. It just depends what the male is going to donate. So the male has an X or a Y, 50-50 in terms of what he will end up, what would end up being in the gamete that fertilizes the egg. So it would be the male that would determine gender based on the 50-50 chance of whether it was the X or the Y chromosome that went into the sperm cell that fertilized the egg. And just note, girls, whenever the guy tries to tell you that he's complex, that he's complicated, look at this tiny little Y chromosome compared with the X chromosome. Don't believe him. Yes, so there's 46 chromosomes in total when you include the homologous pairs, or the other ways there's 23 chromosomes. We could say that labeled 1 to 23, but for each of those numbered ones, there is a pair. There are two of them, one from mum, one from dad, and importantly, this is what they look like before they're duplicated in preparation for mitosis. This kind of X shape would be what we would see when it has been duplicated and mitosis is about to happen. And just keeping in mind, they don't look like this all the time. It's only when they've been duplicated for mitosis. There can be a long time in the cell's lifetime where it's not looking to divide, it's just sitting there doing its job. And if that's the case, we're not going to see the X. It's going to look like what we saw in the previous slide. It's going to look like this one. So the phases of mitosis. So now, just keeping in mind, mitosis is a continuous, gradual process. It's not a process where something happens and then there's a pause, and then something else happens and then there's a pause. It's a continuous process, but we have broken it up into these phases. They make sense and they're identifiable. Now, also keeping in mind with this, now technically if we're talking about everything that happens with cell division, we would talk about these six kind of points that are happening. But technically, when we are talking about mitosis, though, mitosis strictly refers to nuclear division. So what's happening with the nucleus and the nucleus dividing into two. So mitosis concentrates on these 4 phases. And but we still need to, just to have some proper context, know these ones that are at the beginning and the very end. Now, the simple thing to, I guess the simple thing to try to remember that is let's remember PMAT. So in order of how that goes, we've got PMAT. Let's remember PMAT. I'm not sure how you can try to link that in. Try to work out your own way, but PMAT will help you kind of remember the order of the phases. So let's start explaining them from the beginning. So firstly, before we're getting to our PMAT, we'll just look at that first introductory stage, which is interphase, an early prophase. So this is where the chromosome actually is copied. So the chromosome replicates. Now the sister chromatids, which are now identical copies, they're held together by the centromere. We saw that before. So the chromatin, we could just think of that describes both the DNA and packaging proteins together. So normally it's all spread out. It's not yet identifiable as individual chromosomes. So now when we get to prophase, so the P in our PMAT, now technically we've got where mitosis begins. So the chromatin condenses now to form visible chromosomes. So before an interphase, early prophase, looking at the chromosomes, or looking at the DNA under a microscope is kind of a diffuse, kind of spread out. We can't really determine individual ones. It's prophase where we'll start to see individual chromosomes. So we'll start to see them, and then they'll condense even more to a very tight little package. The other couple of things that happen in prophase is the nuclear membrane will start to break down. That's why we've got a dotted line here. And the spindle fibers begin to form. So the spindle fibers, we can think they're like little ropes that are going to start to form. They are going to end up attaching to the centromeres on the chromosome. So just notice they're not there before, but once mitosis starts, we're starting to form these little spindle fibers. Okay, so the next phase for our PMAT is we've just had P, when now we're at M, so metaphase. Now, another thing we could think with metaphase is think metaphase middle, because what happens in metaphase is we have the chromosomes line up in the middle of the cell. So they line up at the center. The spindle fibers that were forming in prophase are now fully formed and attached to the centromeres. So what we have metaphase in middle and spindle fibers forming, and notice the nuclear membrane has now completely broken down. Now anaphase, we can think of anaphase as A and think away, because the main thing that happens with anaphase is that the chromosomes are pulled to opposite, that were lined up in the middle, they're pulled to opposite ends. So the proteins are kind of split at the centromere, splitting the sister chromatids apart into two separate single chromosomes. The spindle fibers are still attached at the centromere, and they will actually pull each single chromosome to opposite sides of the cell. Now, lastly, telophase. So we now have had a situation after anaphase where we've had the chromosomes pulled to opposite sides of the cell. So one side we're going to have some chromosomes, the other side we're going to have chromosomes, and note that on each side we have the same, the DNA that's on each side is the same, because we've just simply split the chromosomes that were the X shape, we've simply split them in half and pulled them to opposite sides. So everything that's gone to one side is exactly the same as what's gone to the other side. So now the spindles fibers, which was the rope that pulled them away, will start to break down. We're going to form a membrane around what will be each new nucleus where the two sets of chromosomes are. So we now have an elongated cell that has kind of two nucleuses. So the actual nuclear division part is complete, but the cell division part isn't complete. The last step is cytokinesis. And looking at the word, it just kind of literally means like cell movement. We're splitting the cell in two, and that's in animal cells. We're simply just kind of like pinching it in the middle. It just kind of pinches in the middle until eventually turns into two cells. So that's quite simple. Now in plant cells, it's a little bit more complicated. That's because plant cells have a cell wall. We can't really pinch it in the middle. So a cell plate forms. We can kind of think of it as like a little membrane kind of starts to form first, and then a whole cell wall kind of forms over that membrane to be forming two cells. Now this just shows you some examples of what these phases will look like under a microscope. So here we have like a plant cell would start looking first. So we've got interphase, we can see that the DNA is spread out. We can't really determine individual chromosomes. Here it doesn't look like we can see kind of clear X's, just because there's lots of chromosomes, but it has condensed to the point where chromosomes are visible if it was a better quality picture. Metaphase, we kind of see that things have lined up in the chromosomes have lined up in the middle. Anaphase, we see that we're starting to kind of pull them away to either side of the cell. Now telophase, we can now have the where the chromosomes are, we're forming two nucleuses. And this can kind of show you what, before, where we had the cell plate would start to form. It's like where we'd have start to form a little membrane across where it's going to divide, then we would form a cell wall there. Now, just an example in animal cells, again, DNA spread out in interphase. We can start to see chromosomes in prophase. Spindle fibers are forming, nuclear membranes breaking down. Metaphase, they're all lined up in the middle. Anaphase, they've been pulled to either side. Telophase, we're then kind of forming membranes and two cells. This is, I guess, a much clearer example. So interphase, we see the DNA is kind of spread out. Here we're kind of showing early mitotic prophase, so that kind of, we can start to see some individual chromosomes forming, but they're not fully condensed or packaged yet. So here where we see the late prophase, we now see some nice tight packaged chromosomes. They're still spread out everywhere though. We get to metaphase, we see that they've kind of lined up. If that was the middle line, they're lined up in the middle. We get to anaphase, we see that they've been pulled to either end, and then telophase will start to form two nucleuses. Now, this is an important slide to remember. It's the last slide, but this is something that we've been doing throughout this topic. We need to relate everything we're doing to continuity of the species. So this is a good kind of model answer to remember, that could get you some marks if you ask a question like this, because it's likely that you would be. So we've got in multicellular organisms, mitosis ensures that there is growth and repair. So this means that the individual organism is kept healthy and functioning, which would allow it to live long enough to have the opportunity to reproduce. So by being able to reproduce, the continuity of the species is ensured. And we've just got, so in unicellular organisms, mitosis actually allows for reproduction. As a new generation of individuals is created, it therefore allows for continuity of the species. So if I were to explain that in a different way, if we're talking about multicellular organisms, and now mitosis is not involved directly in reproduction. We could basically just say that mitosis is just involved in trying to keep the whole animal alive, so that it will be able to reproduce. So we just need normal growth, normal repair happening. Mitosis is important just to keep it alive so it can reproduce, and that's going to be something that will more involve meiosis later. Whereas if in unicellular organisms, mitosis is reproduction. I just wanted to show you some. I'll stop sharing this because that is the last. We've just got a couple. Actually, you just got a couple of videos I can point you to watch. Some links in the PowerPoint, or if you search for those, it's a couple of good videos to watch. Because this stuff involves kind of moving things, seeing some actual animation and stuff moving kind of can help you understand. Now, and so on that note, I'll just cut to here where I've got some really high-tech animation kind of ready for you. If we kind of talk through what would be happening, let's kind of pretend our chromosomes aren't there. So if we were talking talking interphase and things starting off, we would expect to see just a bit of a mush of just DNA spread out everywhere. We wouldn't see individual chromosomes. That would be when the DNA would be replicated, but we wouldn't be able to actually see what the individual chromosomes look like. Once we get to prophase, we would then see the chromosomes condense. We would then be able to see individual chromosomes forming. Now the nucleus, or the nuclear membrane, is going to be breaking down. The spindle fibers are going to be forming, but note that our chromosomes aren't, they're not arranged in the middle. They're kind of just spread out everywhere. So if we let's see if we jump to metaphase, let's kind of assume that now that I guess the whole sheet of paper would be the nucleus, and I've got a little dotted line to show them where they would line up. So we know that they're going to line up in metaphase. Now let's also assume that our two colors are homologous chromosomes. So if we kind of have these two red ones, let's just say this is chromosome 1, for example, and I've got one from mum, one from dad. These ones are chromosome 2. I've got one from mum, one from dad. Now, when they line up, there's nothing that says that they have to be together, or also that the homologous pairs of chromosomes have to be together. So for example, we might, this one might go there, this one could go all the way over there. So what happens with one does not affect the other. So they could line up like that in any order, like that, well, whatever you want to do, but they'll just do their own thing. What happens with one does not affect what happens with the other. So that would be the metaphase. Also, what would have happened in metaphases, the spindle fibers would be fully formed, and they would have attached to the centromeres, which are at the center of the chromosomes. Now, for anaphase, what's going to happen is we are going to cut the chromosomes in half where the centromeres are, and we're going to pull. So just hopefully I don't ruin my high-tech blue tack and paper chromosomes. So we're going to cut them in half, and we're going to pull them away like that. Again, that's going to go like that. That's going to go like that. And that's going to go like that. So we'll have kind of cut them in half, and then that's just going to get pushed to that side. That's just going to get pushed to that side. So note that what we have is an exact copy. So here, for this chromosome, we get the exact copy goes there. The copy goes there, copy goes there. So each new cell is going to get exactly the same DNA. And when they're cut in half and go to the other side, again, it doesn't really matter how they lined up or where they were. Like because they're all just going to get cut in half, and the same DNA that ends up here is going to be ending up in the other cell to be creating two identical cells. That's important to know now, because when we compare it with meiosis, it's going to look a little bit different. That's why I'm trying to highlight this point. And I think with that, I will leave it. Thank you.